Greetings. I'm excited to present our work, GeneticBPE, Motif-Preserving Tokenization for Robust miRNA Modeling. I'm Jaskaran, and this is our joint work with my colleagues Prabhav Sanga and Arun Kumar Dubey. This is part of a submission at the Tokenization Workshop at ICML 2025. Our paper ID is 36. Our focus today is on a small but crucial design choice, tokenization, that can drastically improve generalization in biological sequence modeling. Tokenization plays a foundation role in biological sequence modeling, yet it's often treated as just a mere preprocessing step. Standard methods like BPE and Unigram optimize for frequency or likelihood, but they completely ignore the biological structure. In microRNA, which are short non-coding RNAs that regulate gene expression, we have critical motifs like 6 to 8 base seed regions and conserved stem loops that drive binding specificity. When these get fragmented by naive tokenization methods, we lose crucial biological signal. The model has to learn those broken subpatterns that don't align with the true biochemistry of the sequence. This results in poor generalization, especially when models are transformed across species or used in low data settings. So our core hypothesis is simple. Tokenization should encode domain priors that align with biological structure. Our key idea is to rethink tokenization as more than just text compression. It's an inductive bias for models. In standard BPE, the core idea is the merges are purely frequency driven. So the important motifs get split abruptly across tokens. This means biologically meaningful regions like a seed sequence that must remain intact for miRNA targeting are scattered into subwords that the model struggles to reassemble. This dilutes the signal for downstream tasks like conserved family classification or cross-species prediction. Our solution, genetic BPEs, is derived from the standard BPE, but it explicitly encodes biological knowledge. Known seed motifs, conserved flanking regions, and structural priors are used to guide the merge process. So the tokenizer itself knows when to stop merging to so that it prevents to fragment a motif. Intuitively, we are saying let the tokenizer respect biology so the model can see biologically meaningful jumps. This is a genetic construction pipeline. We start with a corpus of raw data level sequences. In this case, mature miRNAs from MirGeneDB database, and we annotate all the known conserved motifs. This can be seed regions, binding sites, or even structural stem and flanks, and these are stored in a motif bank. Then during the merge process, where every pair is stored not just by frequency like the standard BPE would do, we also it also is affected by whether the merge stays inside a motif or it would cross a motif boundary. So if the merge stays fully inside a motif, it gets a positive bonus score. But if it crosses a motif boundary, it gets a penalty. This is what gives the algorithm the ability to preserve biological integrity. Over multiple iterations, the favored motifs are taken into consideration, and a final vocabulary that aligns well with the known biology is formed. Instead of generic subwords, now the tokenizer will emit tokens that often map to meaningful biological units. The genetic BP algorithm is focused on this exactly. The score of two sequences merging of the frequency count between token A and token B is calculated as the frequency that the standard BP provides. And we include two terms. One is bonus AB and one is penalty. So the bonus counts how many times the pair appears inside a conserved motif span given in the motif bank. And the penalty counts how many times the pair would cross a motif boundary if merged. The parameters we use are lambda and mu, and they help us control how strongly we want to bias towards preserving motifs. A larger lambda would put more weight on keeping merge inside motifs, while a larger mu will strongly discourage merges that would fragment them. We will set the mu to be larger than lambda, ensuring the penalty always outweighs any small frequency gain. So every merge decision tries to balance frequency-driven comprehension with domain-aware motive fidelity. For our experimental setup that we set to evaluate genetic BPE, we designed experiments that test the approach under real biological constraints. So we use particularly MirGeneDB version 3, which includes over 20,000 mature miRNAs over 114 species. So in that we created two sub tasks. One and for that we created sub datasets from the main data. One is Burberry, which is a binary classification. Here the goal was to classify whether a miRNA belongs to a conserved family or not. That is it appearing in two or more species or a non-conserved family? And one was multi-top 50, which was a multi-class classification. Here we predict the top 50 most frequent miRNA families a sequence belongs to, namely MIR31 or MIR219. So then we compare genetic BPE over five baselines: car level tokenization, k-mer, where k was 3 and k was 4, Unigram LM, and standard BPE. All tokenizers was constrained to the same vocabulary size, around 512 tokens, and once trained, they remain frozen for the entire modeling process. So, and for fairness, the downstream model is a standard for vanilla transformer in all scenarios. So the results were quite showed that genetic BPE was quite better as compared to the other tokenization methods. On the Burr binary task, genetic BPE genetic BPE achieves 90.8 accuracy compared to 87.4 for standard BPE, hoping 3.4% improvement. And when we look at the domain shift, training on human miRNAs and testing on zebrafish and fruit fly, the cross domain generalization gap dropped by about 50%. The motif distortion rate was also The motif distortion rate was also a key metric. It is only 0.05 for genetic BPE, meaning we preserve 95% of known motifs compared to 75% for BPE. The takeaway is clear. We don't have to pick efficient compression and biological fidelity. With genetic BPE, we can get both, as the average compression ratio stays around 3.1x. So we reduce sequence length without sacrificing motif integrity. So to conclude and wrap it up, genetic BPE achieves 3.4% higher accuracy than standard BPE on binary miRNA classification. Cross-species generalization gap is 50% lower. Motif distortion rates drop to 0.05, preserving 95% of the motifs. Compression ratios stay high at 3.1x to keep efficiency. And our results showcase that domain-aware tokenization is a powerful inductive bias, and a small tweak like genetic BPE can deliver robust results in biological NLP tokenize pipelines. Thank you for the presentation. All our code, motif files, and supplementary material are available on GitHub, and we hope this inspires more work on domain-informed representational learning, especially in the area of genomics and functional biology. Thank you.